will be giving a quick tutorial about TIMEOR, Trajectory Inference and Mechanism Exploration using Omics data in R. This is the first automated interactive web and command line time series multi-omics pipeline method for differential expression and comprehensive downstream analysis. TIMEOR reconstructs interpretable and user-guided gene regulatory networks, and specifically, TIMEOR's web interface leverages users' knowledge of the biological process by providing options to the users during each analysis phase of these three. TIMEOR first takes in raw sequence reads in the form of FASTQ files for RNA-seq and performs all analysis from quality control and differential expression to then enrichment. TIMEOR then integrates motif and chip data to define transcription factor binding patterns and reconstructs the transcription factor gene regulatory network. This web interface enhances reproducibility and ease for users to dedicate more time to interpreting results and planning follow-up experiments. So I really hope you enjoy, and here's a quick tutorial. So as I said, the first step is to load your raw data, and this can be in the form of either a metadata file directly and then a count matrix, or the SRA run table that you'd get from going to Geo, and this will be constructed into a metadata file, and then all of those FASTQ files that are in this SRA run table will be downloaded for the user. Once this is done, the user needs to answer questions about the experiment. What type of organism? What type of sequencing? What type of experiment? What type of time series data? And then if you want to compare multiple methods for alignment and differential expression, and then you need to answer a question about what the maximum number of time step is over which one gene can influence the transcription of another gene, just so that we know the window of time to look for interactions. And then you click Run. For this tutorial, I'll be describing the two ways of starting from your either raw data or a count matrix. First, we're going to start with real data, which I've loaded for you here, by just clicking on SRA run table and raw data. This will load the SRA run table. I've given the two examples of the FASTQ files here. This SRA run table is converted into a metadata file. The default adaptive parameters are set for you, and then you just have to click Run. The raw data will then be retrieved, quality checked, aligned, and saved as a read count matrix. So these data have been retrieved. The quality control has been completed. You can download interactive results here and look at them. The data have then been aligned in two ways, given that I've said yes to comparing multiple alignment methods. You can choose the one you want and then generate the count matrix. Then this is passed to the next stage. The other way that you can run this is starting with a metadata file that you have already produced and a count matrix. So in that way, you don't have to go through these particular steps, and you can jump right to the next step. So this is our count matrix metadata file, and this is our count matrix. Principal component analysis is performed. We can also correlate our replicates, either using the Spearman or the Pearson correlation, and we see the loadings for the principal component analysis. These data can then be normalized and corrected using multiple normalization methods and the Harman correction method. And again, we see the principal component analysis for the normalized and corrected data, its associated loading plot, and the correlation of replicates. Here I'm showing Pearson. These data are then passed to primary analysis, where differential expression is performed, and we compare multiple methods, as that was what parameter we set in the pre-processing stage here. Once you determine the result folder name and the adjusted p-value threshold, you run, and all of these methods will be run. These resulting differential expression genes can be compared. You can also add a previous study, as I've done here. You can then display the desired differential expression method results that you choose and perform clustering on the gene expression trajectories for that associated method. In this case, I've let TIMEOR automatically cluster, and it chose these three clusters. You can also save your place at any time here. These data are then passed to secondary analysis, where enrichment is performed, followed by factor binding and temporal relations. First, for each cluster that you choose, in this case I'm choosing this red cluster, you can see all of the genes here. You click Analyze, and Go analysis is performed, followed by pathway and network enrichment, as well as motif analysis, and the user can download the interactive meme results to visualize. In factor binding, these gene expression trajectory clusters are then teased apart to show you the observed transcription factors that are perturbed in your data, in addition to the top predicted transcription factors that are formed by a consensus of multiple transcription factor prediction methods. So here you're seeing the observed transcription factors and the top 4 predicted transcription factors that are a consensus of these methods. And if at least 40% of the methods agree on the top predicted transcription factors, then they are listed, otherwise the cell is left blank. The user can then see if there are associated chip data with these transcription factors, their ENCODE IDs are listed. The user can look at the details of individual method prediction for transcription factors for each cluster. So for example, in this cluster 3, these are the associated transcription factors that have been predicted for the top 4. And where there are blanks here, this indicates that the enriched motif is not assigned to a transcription factor region. And in this case, it might be useful to look at the interactive cluster motif results here, which is searchable. Using these ENCODE IDs, the user can then go to ENCODE, download the associated bigwig file of interest, and then load those data to show the average profile of that particular transcription factor over the different clusters. And this can be done for 3 transcription factors at a time. And on the last tab of temporal relations, these observed and top predicted transcription factors are shown in a transcription factor network, and TIMEOR associates these transcription factors, both observed and predicted, using temporal relation information from the gene expression trajectory clusters to show 4 types of relationships, known interactions that have been experimentally determined between two observed transcription factors, 1 predicted on an observed transcription factor, and then there are also predicted interactions between the predicted and observed transcription factors, in addition to between 2 observed transcription factors. These information altogether can then be displayed as the user sees fit by moving genes around, as TIMEOR has StringDB embedded within it. All of this information is available in the tutorials section of TIMEOR, and these results can be downloaded by just clicking on the download results page.